Welcome, new trainees, to Orientation Day. Today, you're stepping into one of the absolute most complex, highly coordinated, and frankly, awe-inspiring facilities in the known universe. Now, in this explainer, we aren't just going to look at microscopic biology the way a textbook does. Instead, we're going to tour the cell as if it were a massive, bustling city factory. We are going to explore the transport highways, the specialized workstations, the structural foundations, and the flexible boundary walls that literally keep this entire production facility running at top speed. So here is our floor plan for today's tour. We'll kick things off with a welcome overview, head right into the assembly workstations, check out the logistics highways, do a quick structural safety inspection, examine the outer walls, and then we'll wrap up with a mandatory emergency drill and trainee quiz. Let's dive in. Section 1. Welcome to the factory. Let's get an overview of this site. As we zoom down onto the bustling factory floor, the very first thing you need to understand is that nothing, and I mean nothing, here is static. This whole facility is a non-stop hive of activity. Thinking about the master blueprint, you can really see how perfectly all these components interact. Down at the base, we're securely anchored to our foundation. Running vertically, you've got these massive logistics highways called microtubules, managed by specialized centers like centrosomes. Weaving through the cytoplasm are these deep structural anchors known as intermediate filaments. And right up top, forming specialized structures like microvilli, are the dynamic actin microfilaments. It's this brilliantly orchestrated layout entirely designed to maintain optimal workflow. Section 2, Assembly Workstations. This is where we create our essential products.
Now, obviously, no factory functions without an assembly line, so let's walk over to our primary workstations, the ribosomes. These supramolecular particles are the actual machines responsible for manufacturing our products. And the sheer efficiency here is incredible. A single ribosome is made of ribosomal RNA and proteins, and it's divided perfectly into a small subunit and a large subunit. But our factory doesn't just use one machine at a time. Oh no. Multiple ribosomes attach along a single instruction line, the mRNA, creating this massive, high-volume polyribosome production line. That assembly of small and large subunits directly dictates our total production capacity, just churning out proteins back to back.
We actually split these workstations into two main divisions on the floor. First up, we have the free ribosomes. Think of these guys as the machines that build the internal tools used strictly within the factory itself. So they synthesize proteins for the cytoplasm, the nucleus, the mitochondria, and peroxisomes. Then we have the bound ribosomes. These are physically attached to our internal membranes, and their entire job is manufacturing parts for export or very specific storage. They're building proteins for the cell membrane, lysosomes, or secretory proteins that are destined to just leave the factory entirely. Section 3, Logistics and Transport. Let's talk about microtubules, our internal transport system. So once a product is manufactured, how does it actually get to the loading dock? That's where our internal highway routes come in, carrying heavy cargo like secretory vesicles straight to the plasma membrane. But these highways aren't just, poured concrete. They are dynamically constructed modular tubes. Step 1, alpha-tubulin and beta-tubulin proteins combine to form a basic molecular building block. Step 2, these blocks link up end-to-end to form a single continuous track called a protofilament. And step 3, exactly 13 of these small specialized tracks align in a circle to form a complete, sturdy microtube. And a fun fact, these tubes don't just route cargo, they actually organize the massive division of the factory into two parts during cell division by forming the mitotic spindle. Pretty wild, right? Section 4, Structural Safety and Stability. Time for the inspector's report. Okay, grab your hard hats, trainees. With all this heavy machinery and moving cargo, you're probably wondering how the building doesn't just collapse on itself. Well, the secret lies in our intermediate filaments. We don't just use single, weak threads. We use a heterogeneous protein family that tightly coils together. They start by forming dimers, then staggered tetramers, and eventually they lock together into a massive lateral association of 8 tetramers. What you get is this heavy-duty, staggering structural cable that is just incredibly resistant to tension and physical stress. It's the ultimate structural stability for the factory. So the really crucial takeaway here is that these intermediate filaments serve as our ultimate anchors. They don't just hold our internal shape together, they literally build strong connections between neighboring factory buildings through cell-cell junctions. That ensures the entire industrial park stays completely stable. Plus, they form the nuclear lamina, which builds this impenetrable safety cage right around the factory management office, the nucleus.
Section 5, Outer Walls and Locomotion. Time to look at our flexible building materials. A factory can't just be purely rigid, right? It has to adapt. Sometimes our city factory even needs to crawl across the terrain. And this is where the protein actin comes into play. Actin filaments are our multi-purpose flexible scaffolding. They form two-dimensional networks and three-dimensional gels that are super concentrated in the cortex, right beneath the plasma membrane. That support grid fundamentally reinforces the outer boundary of the factory. But amazingly, they also drive whole cell locomotion, physically helping the factory move across the terrain. On top of that, they assist in pinching the factory in half during cell division and even help transport organelles near the perimeter. The way this dynamic building material works in real time is honestly brilliant. Whenever the factory needs to push a wall forward to move, free actin monomers just snap right onto the plus end of the actin filament. It rapidly extends the structural support in a very specific direction. It is literally like scaffolding that magically builds itself precisely where and when you need to expand the building. Section 6. Emergency Drill and Trainee Quiz. Let's test the systems. Attention on the floor. The safety alarm is sounding. Let's see what you trainees have actually learned about our structural roles and hazard protocols. First, imagine an emergency scenario, a severe OSHA violation on the factory floor. What happens when transport tracks fail? Well, if a microtubule were built of just a single protofilament, it would be extremely thermally unstable. Just a single bond breaking in the middle would cause catastrophic track failure. But because our factory mandates multiple protofilaments, 13 to be exact, breaking the tube in two requires breaking multiple bonds all at the exact same time. This thermal stability is what prevents disastrous accidents from shutting down our logistics. All right, rapid fire quiz time. Question 1. Which factory workstations are responsible for manufacturing tools used strictly within the factory floor rather than making parts for export? If you said free ribosomes, you are absolutely correct. These supramolecular particles act as our internal assembly lines, synthesizing the vital proteins needed for the cytoplasm, the nucleus, and the mitochondria. Okay, moving right along to our second emergency drill question. What flexible building materials create the two-dimensional and three-dimensional support grids that reinforce the factory's outer boundary and allow for whole cell locomotion? And the answer is actin filaments. These dynamic linear bundles form gels and networks right in the cortex beneath the plasma membrane. They're adaptable city walls that protect us and let the whole factory migrate when it needs to. Final question. Let's satisfy the safety inspector. Question 3. What heavy-duty cables build strong structural anchors between our facility and neighboring factory buildings to maintain park-wide stability? That would be our intermediate filaments. By forming specialized cell-cell junctions, these staggered, coiled proteins make sure our foundation remains locked perfectly to our neighbors, completely preventing structural collapse under mechanical stress.
Well, congratulations, you've passed orientation with flying colors. But before we go, as you look out over this incredible microscopic factory and see the sheer coordinated precision of the ribosomes, the microtubules, the actin, and the intermediate filaments, I want to leave you with a provocative thought. If all of these intricate scaffolding networks and specialized workstations depend on absolute unwavering perfection, what kind of cascading failures occur when just one single assembly line encounters a genetic mutation? Think about that next time you ponder the blueprint of life. Thank you for joining this explainer, and stay curious.